actually joining from the Eastern Hemisphere. So can everyone hear me clearly? It's all good, right? Can you guys hear me?
Yeah, that's better. Yeah.
Okay, perfect. So I'm Thilina. So this throughout my presentation, I would like to highlight some of the eDNA studies we are doing in Shushanbana, China. So in the previous talk, also talk about the eDNA. So here my talk is more only focusing on the eDNA, which is subclass of the environmental DNA. So we call it like eDNA, which is an invertebrate derived DNA. So there we take the materials from the invertebrates itself, and then we'll use that material to get an information about their trophic ecology, or maybe use that one for the biodiversity monitoring. So when it's come for the dung beetle, recent studies are actually used dung beetles as one of the eDNA sampler for the biodiversity monitoring and the ecological studies. There are three groups of dung beetles. The first one is like the Scarabinae, Iphidae, and Geotriphidae subfamilies. So in the Scarabinae, known as the true dung beetle because of their highly association with the mammalian feces. So they use mammalian feces to as a food source and also to make their brood ball for their larvae. So based on their food relocation behavior, there are three types of dung beetle. The first one, they make the dung ball and bury beneath the dung pad. And the second group we call rollers or the telecoprids. They make the dung ball and roll away and bury somewhere else. And when it's come for the third group, they actually live inside the dung pad. So recent studies, as I mentioned, they use the dung beetle as a sampler to do the biodiversity monitoring for the mammals or the vertebrates. However, the dung beetles also are actually associated with the fungi. So the dung beetles are known to feed descending from an ancestor which actually had the fungi as a main dietary source in their trophic ecology. So therefore, fungi is one of their alternative food source they actually feed on when the large mammals are locally extinct. So in the current studies I'm going to present, we actually look at the dung beetle in the Shushanbana Tropical Botanical Garden, Shushanbana Tropical Rainforest, and the adjacent rubber plantation. Why we selected the rubber plantation is, the rubber plantation is the main habitat conversion in the region. So this is one of the main threat for the forest loss, cause for the forest loss in the area. And we collect the dung beetle from the dry season and also from the rainy season. So there we used 20 hectare plots, and each 20 hectare plot, we deployed 5 1 hectare plots. So within each 1 hectare plot, we deployed one pitfall trap baited with the human feces with the preservative solution of the propylene glycol and ethanol. And also we collected some vegetation data, some soil properties, and also climatic data for the subsequent analysis. And for the three studies I'm going to talk today here, I selected 3 dung beetle species. Those were widely distributed between rainforest and the rubber plantation, across rainy season and across the rainy season and the dry season. So therefore, we actually could be able to control the effect of the species on their gut content. For the first study, we look at the mammal, we collect the dung beetle from the rainforest during the rainy season, and we're looking at the mammalian composition using the 16S gene. And the second study, we also include the rubber plantation and the dry season. There we look at the ITS targeting the fungi from the dung beetle gut. And the third study, we incorporate the bacteria from the dung beetle gut using the metabarcoding. So all this analysis, bioinformatics were conducted using data to pipeline in R, and the subsequent analysis done in R. So in the below, I'm showing the gut extraction for the dung beetle using the 3-step method proposed by Gomez. So there we try to minimize the contamination from the environment, fungi spores or the other bacteria coming from the environment. For the first study, what we look at is actually to enhance the existing method to use the dung beetle as a sampler to monitor the mammalian community. So the recent studies, they use the dung beetle as a sampler to look at the mammalian, their mammalian meal source. And however, and also they use that one to sample the mammal. So however, all these studies, they use the live beetles. There are some caveat when it's come for the live beetles. They use the hand collection. So when you do the hand collection for the dung beetles, there are some taxonomic bias. Generally, when they do the hand collection, they are focusing on the dung pads. So in the dung pads, you only can find the endocropids, which are the dwellers. So they basically ignore the rollers and the tunnelers. And also there's a higher chance of the losing DNA when they use a live pitfall trap with the water as the preservative solution. However, the most commonly used method is to use the pitfall trap with the preservative solution to collect dung beetles. They can actually capture a representative sample from the community using the baited pitfall trap with the preservative solution. However, until now, it is unknown whether we can get the DNA from this traps. So in this project, we are looking at whether the dung beetle collected from the conventional baited pitfall trap with the preservative solution does actually preserve the dung beetle gut content. For the analysis, we listed the existing mammal species in Shushanbana. Then we extracted their sequences from the NCBI, and we extracted the 16S gene using the genus prime. Then we look at the mammal composition in the dung beetle gut. So then there we found we used like 18 dung beetle individuals for this study, and they are the paracropids or the tunnelers show the highest sequence richness. There are total, we found around 8 non-human mammals. There we exclude the human sequences from the analysis. There we found the common species like the wild boar, and also the rare species like Asian black bear, which did not find from the camera trap studies done in the particular study site. Out of the 8 mammals, the 7 was found from the tunnel species. So this is the very first study that showed that the dung beetle collected from the preservatives using the baited pitfall trap with the preservative solution can actually get the mammalian dung beetle mammalian meal source. There we found the rare species and also common species. However, the least number of species were recorded from the rollers, which makes sense because they are mostly rely on the single source of dung to make their brood bowl and also for their diet. However, when it's come for the paracropids, they are known to process the dung processed by the other rollers or the dwellers. So therefore, we could able to capture the highest number of species from the tunnelers. So this method is given an additional tool to sample the mammals. And also the most commonly used trap type in the dung beetle studies are the one actually we described now. So if we incorporate the eDNA for such ecological studies, actually we can look at how the dung beetle and the mammal interactions happens. There's a caveat in our study, which is actually we didn't we didn't compare the live versus our method. However, this is like a one simple and also the one of the cost-effective method we can use to sample mammal species. So let's go to this. When you go for the second study, this is under this first revision in the molecular ecology resources now. So there we actually looked whether the dung beetle eDNA from the dung beetle gut can use to sample the fungi. It is known that all these biodiversity frameworks, they highlighted the species monitoring, because the species monitoring or the biodiversity monitoring is crucial to make the conservation decision and implement the conservation decisions. However, it's kind of tricky and difficult when it's come for the taxa-like fungi, because there's a huge number of species in the fungal community, and there's a small fraction of the species are described up to now. When you use the conventional sampling for the fungi, you go to the field and collect the fruiting body, and you grow that one in the agar or other media to look at the species. So that one take long time. For an example, this study, they collected the sample, the fungi for over 200 weeks. They only could able to extract like 200 species, which is kind of low compared with their total diversity. So we assume that this could be actually compensate, or like this actually could help by the eDNA technique or the subclass of the iDNA, because iDNA can capture the small fungal spores and also viruses such as like small entities like the viruses from the species guts. Recent studies, they use the iDNA not only for the biodiversity monitoring, but also they use that one for the measure the conservation outcomes. So in this study, we looked at, as the first question, whether we can use the dung beetle iDNA to sample the mammal community in a given habitat. There we hypothesize that the dung beetle may feed on the fungi community in the rainforest. If you look at the dung beetle gut, maybe we could be able to find this particular fungi community. Second, we hypothesize that the season may play an important role in this scenario. Therefore, in the rainy season, it's the moisture availability is high for the fungi, and the fungi growth will be enhanced, that there will be a higher diversity of the fungi, which we can potentially get from the dung beetle gut. And the last, we assume the fungi community may be affected from the habitat conversion from the rainforest into the rubber plantation. So therefore, the available fungi for the dung beetle to feed on is going to be really low. Therefore, the fungi we can find from the dung beetle gut may be also low. So this method, possibly we can capture the habitat effect of the anthropogenic habitat conversion on the fungi community. So there, for the analysis framework, first we look at the fungal composition, and then we assess the sample completeness. Then we look at the fungi diversity from the dung beetle gut, and then we look at whether there's an effect of the season or the habitat or the species. And the last, we look at the community composition, and also we look at how the community variability happen in each habitat. For the fungal community composition, we found over 9,000 fungal taxonomic unit ASCs, and belongs to the 500 genera, and belongs to the 12 phyla. So when you look at the composition, the Basidiomycota is getting higher during the rainy season. However, in the dry season rainforest and also the dry season rubber plantation, the Ascomycota is getting more dominant. For the when you look at the diversity, the rainy season rainforest showed highest number of sequences for the based on the total ASCs, and also the ASCs or the sequences identified at the species level. When you look at the column in the right and the column in the left, there's a huge difference, more than 30 times different of the species identified from the known database and the total ASCs we found. So it's clearly showing when it's the one actually pointed here is the rainy season rainforest is showing like a higher diversity compared with the dry season rainforest and the rainy season rubber plantation and the dry season rubber plantation. So when it's come for the diversity, so the diversity doesn't change across between the species. However, in the rainy season rainforest, the fungal diversity found through the dung beetle gut is significantly higher. When it's come for the community variability, it's more driven by the turnover rather than the nestedness. And the community composition, it's shown that the dung beetle, the fungi we found through the dung beetle gut have the higher, like a completely different community composition in the rainy season rainforest compared with all the other habitat and the season combinations. So when we looking at our results, this is the very first study looked at the potential to use the dung beetle iDNA to use as a sampler for the fungi. There, we found the higher cryptic diversity of the fungi in the rainforest, and also we found that the fungi are actually affected by the habitat conversion, which is actually we could able to look when we're looking at the dung beetle gut. So we found lots of taxonomic units like amplicon sequence variants. However, the one actually we identified into the species level is really low. So it's highly highlighting that the underexplore nature of the fungal community in the given habitat. Also, we done like a qualitative comparison between the existing eDNA method to monitor the fungi using the soil-based eDNA studies. When we compare with the soil-based study and our eDNA study, both studies from like previously published studies, both found the similar fungal diversity in the both habitat and the season. When it's come for the dung beetle eDNA, we use like a single beetle specimen rather than pooling in together to get this higher diversity. However, when it's come for the soil samples, they collect the multiple soil sample and combine them into one sample to get this higher number of fungal communities. Both studies found like a completely different community composition from the rainforest and the rubber plantations. And also, they also found higher Basidiomycota relative abundance in the rainforest compared with the rubber plantation. It makes sense because the Basidiomycota is known to sensitive for the chemical usage. So in the rubber plantation, they use nitrogen-based urea as a fertilizer, which is negatively affecting to the relative abundance of the Basidiomycota. And also the substrate conditions, because Basidiomycota are known to be like an exomycorrhizal fungi. So in the rainforest, have higher diversity of the plants. So therefore, it provides large number and also the variety of the substrate where the fungal can grow compared with the rubber plantation. However, our study, eDNA-based study, found a seasonal variation. When it's come for the eDNA-based, so the soil-based fungi studies, they didn't find in the same habitat or the same locations, they didn't find any seasonal variation in the rainforest. So our study finding is actually aligned with the known ecology or the known behavior of the fungi, where they are more responsible for the climatic seasonality or the response for the rain availability. So when we go back to our first question, we asked like whether the dung beetle eDNA can be used to sample fungi. So according to our first use dung beetle as a tool to survey the hyperdiverse mycological communities in the tropical rainforest. And the second, we found also there's a seasonal effect on the detection of the fungal community, highlighting that if we want to get the highest number of fungal community in a given habitat, we have to consider the season and collect throughout the seasons. And the third, we hypothesize that the dung beetle eDNA actually can capture the anthropogenic habitat effect of the anthropogenic habitat conversion on the dung fungal community, which actually throughout our study, we could be able to detect. Okay, so let's go to the third study. So there we actually look at if the dung beetle can change their diet throughout the season or the habitat, how the bacteria finding from the dung beetle gut can help for their dietary changes. So studies use the microevolutionary process to look at the macroevolutionary pattern. For an example, if you look at the gut microbial composition of a species throughout their dietary gradient, we can actually understand how the species adapted for different types of foods. So this is like microbes an essential component of a species biology, because that one is help a species to adapt to a new food source and also to adapt to a new types of environment. For an example, one of the study done in US, they look at the gut microbes content of a worm species, which actually adapted to survive in a crop rotation, and also the found in the wild types. So they found the species adapted to the crop rotation have a completely different types of gut bacteria compared with the wild types. So it's indicate that when the human intervene happen, so the species actually can adapt to survive for such a situation. It goes with the same scenario when it's come for the dung beetles. So the Australian dung beetles are also known to feed on the native mammal species, and also some of the species are known to feeding on the fungi in Australia. So they found the species are feeding on the dung, and also the species feeding on the mushroom have completely different gut bacterial composition. So given that information, we want to look at whether the dung beetle, we want to look at if does the dung beetle show the different spatial and the temporal dynamic of their alternative food sources, which is the fungi in this case. So then there we assume the dung beetle can feed on the different types of fungi in the different season. And the second, so in the rubber plantation, the available of the substrates are limited, and also it's affected by the chemical usage in the rubber plantation. Therefore, the fungal community may be not much compared with the rubber plantation rainforest, and there may be not a seasonal variation. And the second, we ask the question, if the dung beetle shows such difference in the fungal consumption, whether this one is facilitated by the bacteria. There we assume the dung beetle who actually feeding on the similar types of the fungal community may have the similar types of bacteria, where when they shift into a different types of fungal community, they show the different types of bacteria. And the third, we ask the question, if the dung beetle can change their bacteria and the fungi accordingly, which are the factors are driving this association? So there we assume the species symbiosis, the bacteria and the fungi symbiosis is the main factor driving this co-occurrence rather than the seasonal or the climatic factors. Similarly, we look at the fungal and the bacteria composition, and then we look at the effect of the season and the habitat and the effect of the species for the both bacteria and fungi. And then third, we looked at the fungal and the bacterial composition. And finally, we used the species distribution modeling using the HMSC package to look whether this species co-occurrence is driven by the species symbiosis or the environment. When it's come for the fungi, which I mentioned in the previous slides as well. So the fungi showing the different composition in the rainy season, dry season, and the rainy season. So when it's come for the dry season rainforest and also the dry season rubber plantation, the Ascomycota is getting more dominant. However, when it's come for the rainy season rainforest, the Basidiomycota is getting increased. When you look at the taxonomy, the rainy season rainforest show higher fungal taxonomy compared with the other habitat and the season combination. When it's come for the bacteria composition also a relative abundance also show a kind of a similar pattern. So when it's come for the rainy season rainforest and the dry season rainforest and the dry season rubber plantation, the Firmicutes are getting more dominant. However, in the rainy season rainforest, proteobacteria are getting more dominant compared with the other habitat and the seasons. When you look at the diversities, unlikely the fungi, the bacteria doesn't show any clear pattern, even though the habitat show a significant effect. When you look at the composition, again, the rainy season rainforest show a different composition when it comes for the fungi, even though in the bacteria, the diversity, even though the diversity doesn't change, the composition in the rainy season rainforest completely different from the other habitat and the season combination. When you look at the co-occurrence, it shows the bacteria and the fungi co-occurrence are highly correlated. When you look at the HMSC or the joint species distribution modeling results, when you look at the null model, which doesn't include any environmental parameters or the factors, it shows the fungi and the bacteria have a higher association. However, when we include the environmental factors, all these fungi to bacteria associations are completely disappearing. When we look at the both variation partitioning for the bacteria and the fungi, the main variation was mainly explained by the vegetation or the temperature or the soil parameters rather than the random effect, which is assumed to be the species association. When you look at the explanatory power of the all these developed models, the explanatory power is also increasing when it's come for the full model, which including all the environmental parameters. Unconditional predictive power, again, show higher value for the bacteria than the fungi. Among them, the most important one is the conditional predictive power, which actually used to describe the occurrence of one species or the group, in this case, bacteria or the fungi, how much is explained when the other group is presenting. So here it's showing that the points were higher for the fungi when they account for the occurrence of the bacteria. So therefore, it's clearly highlighting that the occurrence of the fungi is mostly driven by the occurrence of the bacteria. So when we look at the our results, so this is the very first study looking like whether when the dung beetle fungal component is changing across the spatial temporal scale, how it's actually aided by the bacterial changes. The fungi composition and also the bacteria composition is different, and also higher in the rainy season rainforest. And we assume that the microbe turnover is facilitating dung beetle for such changes in the fungal community. When it's come for the fumicultures, is higher in the dry season, both habitat, and the rainy season rubber plantation, which is known to harvest the energy from the structural carbohydrate-based materials. So therefore, we can predict maybe when the fungal diversity is low in those time or the seasons, maybe the dung beetle feeding the other alternative food sources, maybe like the decaying material, which actually the bacteria help them to digest. When it's come for the dry season, rainy season rainforest, the proteobacteria is getting increased, which is those are actually known to digest or produce the energy using the protein sources. And the highest relative abundance species, the Basidiomycota, the fungi species, are also known to contain higher amount of protein. So the other question is arising, if the dung beetle can shift their bacteria composition according to their fungal food source, where actually they get this bacterial community. So it's known that the dung beetle get the bacteria mainly from their mammalian food source, which is during their ontogeny, during when they are making the brood bowl. So the brood bowl is made from either cow or maybe the pig. So the pig and the cow feces are known to have higher concentration of the fumicultures or the proteobacteria. So depending on the availability of the fungi food source, they potentially might change the availability of the bacterial community. So as I mentioned earlier, the species associations were really weak. So it's clearly showing this species association is mainly driven by the season and the habitat rather than the species to species symbiosis. So we found like the bacteria occurrence is mainly describing the occurrence of the fungi rather than the vice versa. It's clearly given an idea how this mechanism or the bacteria and the fungi changes happen. So there we assume maybe during the rainy season, during the from rainy season to dry season to rainy season, so dung beetle adapt their gut microbes to adapt for the different availability of the food sources in the rainforest. However, this is a different case when it's come for the rubber plantation. So it's negatively affected for the fungal community. It have a cascading effect to have a constant and a less diverse bacteria community in the dung beetle gut in the rubber plantation. So we asked the question whether the dung beetle show a different composition or different spatial temporal dynamic in the fungal food component. As we hypothesized, we found actually there's a fluctuation in the gut fungi in the dung beetle gut. And the second, we asked the question whether the bacteria help for such diversion or are they actually co-occur. We found a similar pattern like the fungi for the bacteria as well. However, for the third hypothesis, we hypothesized that this co-occurrence is mainly driven by the symbiosis. But according to our results, we found this rather than the species co-occurrence, habitat use and the season is mainly driven this fungi bacteria co-occurrence. So thank you very much for the listening. So this project is mainly supervised by Professor Akihiro and Professor Ahimsa with the collaboration of Professor Fang Huang. And in our study, we first found like there's the most the most widely used pitfall trap actually can use for the dung beetle mammal, look at the dung beetle mammal interaction and the dung beetle to use for the biodiversity monitoring. And the second we used also the dung beetle eDNA can be one of the tool to assess the Wallachian shortfall in the fungal communities. And the fourth we found third we found the bacteria actually facilitating for the dung beetle to diet shift, fungal diet shift in the rainforest and in the rainforest across the seasons. And if any questions or further clarification, you guys can contact through this email. Thank you very much.